Hello? Yeah, great. Okay, let's slide change your thing. Okay, some of you may be sorry to see me back here. we'll, we'll make the best of things. So I'm going to continue where I left off. I'm sorry I couldn't be here on Friday. But if you'll remember, what we had gone through in the first few lectures is first uh, kind of fluxes through metabolic reactions, particularly glycolysis. And then I gave you a little example of ongoing research where we saw cells that started off in highly gluconeogenic environments where there's limited glucose. These were yeast cells. And as they did gluconeogenesis, they started eventually putting out a resource called trehalose. As that built up in the environment, uh, some cells started consuming this and shifted to a glycolytic regime, right? And that was the beginning point of then utilizing a produced resource. This was something that was not there in the environment before these cells started making it. Through this gluconeogenic pathway, they made it, built it up, and then they started consuming it. And then you had states of cells that were trapped in either state. One was highly glycolytic, consuming this resource that's being put out, while the other cells were trapped as producers of this. So if a more stylized model in 2D or 3D would be something like this, a few cells that then became a large mass of cells making lots of these resources using gluconeogenesis. And then some cells started consuming it, and then you had this two-state system of highly gluconeogenic cells making the resource and other cells that were consuming it, doing glycolysis and doing wonderful things with it. So in general, what we were trying to look at are these three ideas. Of course, after you start looking at a metabolic network, you start to see where you find bottlenecks. And then you start thinking about reactions or pathways that are mutually incompatible. When you say incompatible, it basically means due to chemical and thermodynamic reasons, the two reactions cannot essentially happen at the same time at the same place. And we need to find more and more examples of this to illustrate this idea of how you need to separate biochemical pathways, sometimes even within a cell, so that you can carry out opposing reactions. The other idea we're going to explore today are alternate ways to get to the same destination. Because when you look at the metabolic network, what you realize is that you have this key metabolite, let's say it's at a node. But what you'll already appreciate or see is that sometimes there are many different ways to make it and many different ways to break it down. Some of it comes from what the products of these breakdowns are or new reactions are, right? You're trying to make, say, lipids versus enter the TCA cycle, you will use acetyl-CoA differently. Some of it is also this idea of getting around incompatible reactions. So the example we had in glycolysis and gluconeogenesis, for example, is that PEP is a high energy molecule. It becomes pyruvate, and it becomes pyruvate and donates its phosphate to make ATP, which means then you can't use ATP easily to put that phosphate back on pyruvate to get PEP, because we were looking at where they were on that energy landscape, and PEP is at a much higher energy state than ATP, so you can't transfer from ATP to PEP, which means you have to get alternate ways to go around to make PEP. The way cells do that is in gluconeogenesis, they go from pyruvate all the way around to oxaloacetate and then back to PEP. So there are alternate ways to get to a destination. Now, in a cell at a given point in time, we can actually start to predict or think about which pathway or which flux will dominate. We'll try to explore this through some recent research that we're doing in the lab that stumbled across some of these processes. And finally, how are unfavorable reactions sustained? Okay, and so as always, I'd appreciate talks at questions at any point of time in the talk so that some of these concepts become very clear. Okay, so we come back to this almost a toy model of two states. This is a real wild yeast colony, but they're nicely carrying out these two opposing processes. And now we can ask what makes such a system of two specialized cells possible, right? And what we know from these cells growing in this environment is that these are cells that are being grown in complex media. The only thing that's not there, or not a lot of it is there, is glucose. They have a whole bunch of other things. So of course, the question arises, what drives this gluconeogenic flux and why is it so high? We know this is the wild type complex colony and it has these lots of these light glycolytic cells. I'm going to use light cells and dark cells because it's just convenient for a talk. So you just remember dark cells are gluconeogenic, light cells are glycolytic. These wild type cells have lots of these glycolytic cells. And if you now knock out or block the ability to do gluconeogenesis, you still get a colony. They're doing something, they're alive. They're actually a little smaller. These are not perfectly scaled. But what you don't see are these glycolytic cells. So this shows that the gluconeogenic output is required to sustain the glycolytic cell. That's all there is, right? You still have living cells though. So what drives this flux through gluconeogenesis? Of course, this is the idea that you can carry out gluconeogenesis using various different types of substrates. In theory, you could do this by using small um, 3-carbon sources, or you could have a lot of some fatty acid breakdown products, or you could use amino acids. And essentially what we can see here is if you take a wild type cell without, in absolute minimal media, and you supplement amino acids, you restore all these glycolytic cells at full complexity. What you can also see now is that just one amino acid can essentially completely restore or rescue the colony. Okay, so what you need to do again is to get back to gluconeogenesis. Find different ways to make glucose 6-phosphate. What I'm saying here is there are many ways to enter this point. In the cell, in theory, you could be doing any of these. You could enter from here, something could enter from here, something could enter from here, something could enter from here. It'll all work its way backwards to glucose 6-phosphate and go to trihalose. Here what we can see is just one amino acid can completely rescue this phenomenon. Essentially what's happening is this. Aspartate can convert to oxaloacetate. You have oxaloacetate. You're right here. Oxaloacetate is a nice way to go back to PEP and drive gluconeogenesis. This is exactly what we're seeing. So the conclusion in this is, in this case, these cells have decided to maximally utilize aspartate to drive gluconeogenic flux and create the two-state community.
I have a very quick question here. Yeah, so the uptake of glucose into yeast cells, are they through diffusion or there are glucose transporters as well?
In all cells have glucose transporters. Yeast, of course, have a whole bunch of them, different ones, constitutively very highly expressed.
Right.
So glucose is literally instantly taken up because the surface of the cell is covered with glucose transporters. Right. That's their favorite carbon source. Okay. So even if there's a tiny amount of glucose, it'll be taken up instantly.
Okay, is there like the types of glucose transporters that are expressed? Is there any particular type which is majorly expressed in yeast?
Yeah. So there are two types of transporters. One is a very low affinity transporter. That's the one that's very abundant and present all over the place. The other one is a very high affinity transporter. I mean, a family of very high affinity transporters. Those are highly expressed as you drop in glucose concentration. So now in this case, the low affinity transporters are really high, as well as the high affinity ones which are constitutively expressed. They're all there.
The names are there as an
HXK.
Which one?
HXT.
Okay.
Hexose transporters, and hexokinase is the first step of glycolysis. Okay.
I see. I was just trying to.
So HXT 1, 2, 3, so on.
Okay, I was trying to draw the correlation with mammalian cells where there are, I think, 14 glutes, 1 to 14.
Yeah, so these are just like the glutes.
Okay, so the point was that in mammalian cells, the uptake of glucose by glutes is also maintained by insulin. So the insulin is like the key which sort of unlocks the glute and that takes up glucose. So there is a regulation there between insulin and glucose. So I was wondering, is there any similar thing that might happen?
So there is regulation. It's not by growth factors. Growth factors came much later in evolution. But there are two-step regulations. The low affinity ones are constitutively expressed. Those are not, they're very highly transcribed and they don't have repression. The low affinity ones are controlled by a complex transcriptional loop, and it's not very different in the end from what insulin achieves. Okay, so there's a large family similar to glutes. You have the HXTs, HXT1, 2, 3, and
Thank you.
okay. So now though, the point I want to make in this is, now when I show you something like this, sometimes you might still think that a metabolite has a linear fate. So here I'm saying aspartate rescues these colonies. And the way this would be happening is something like this. Aspartate, oxaloacetic acid can go through gluconeogenesis to make trehalose, right? But we also realize something else. Something like aspartate is a very powerful kind of a metabolic resource because these are both carbon and nitrogen rich. Okay. So now in this case, our hypothesis was that this is driving gluconeogenesis to provide the carbon exclusively for this arm of carbon metabolism. Other things are doing other things. So for example, the mitochondria is still carrying out respiration for ATP and so on. This is decoupled from that. But what we also realize is something like aspartic acid is equally important or useful as a nitrogen donor. So for example, aspartic acid, if you're primarily using this carbon for gluconeogenesis, you're also using the nitrogen from it to make nucleotides. And while we were thinking of these two-state systems and the division of metabolic function, we thought maybe then the relative importance of aspartic acid could be the fact that it is primarily being used for carbon in this gluconeogenic cell, but primarily helping make nucleotides in the glycolytic cells. I've actually miscolored this. It should have been red and this should have been green. My bad. Now, how would you do this experimentally? How would you test this experimentally? The way we would do this, I just wanted to highlight what kind of experiments you can do here, is to now in the system exogenously provide labeled aspartic acid in the same kind of a pulse chase kind of an experiment. Carbon is labeled or nitrogen is labeled, and track the relative fates of these in these gluconeogenic or glycolytic cells. So essentially what we're talking about is this idea of metabolic budgeting. The same resource can be used slightly differently in different cells to result in different outcomes. Even before we did experiments, we could now model this. If you remember what I talked about last week, we had a very simple coarse-grained model. That was before we knew trehalose and aspartic acid and all of that. Coarse-grained model originally only said some resources made and being consumed, and you created a producer-consumer kind of a system. Now we have what we call an agent-based model. This is a slightly better model because now we know there are these specific entities doing these specific kind of things. In this case, now we explore this idea of carbon and nitrogen budgeting. Same molecule, but using relatively for different purposes, right? So we have the same two-state kind of a system where you have some yield of aspartate being converted to trehalose versus not being converted to trehalose, and see if that can control the switching or the formation of the colony as it expands. We always start with some small colony which is fully gluconeogenic because experimentally and otherwise we know that that has to come first. When we do this,
I had a small doubt.
Yeah.
Here.
Way back.
No.
Oh. Where is this echo?
Here.
Oh, okay.
Yeah. Can you just remind me, like,
I'm
before
just hearing
adding,
an echo over here. I don't know. Yeah.
Just before adding the aspartate, can you just remind me, like, what was there in the media?
The media is just low glucose, but it's complex media. So it has peptone, it has breakdown products of amino acids, it'll have some lipid, it'll have it.
So it's the same thing that was being used before also.
Yes. So we're still doing that, but we haven't done that experiment. This is just a model. You can see though how the model predicts how the colony should develop if you now include this idea of budgeting. You see it developing, and then you start seeing the emergence of different types of cells, and then the expansion of the light cells that come out. So this model did remarkably well in recapitulating what we saw experimentally without trying to force the experimental ideas onto the model. So again, I just wanted to highlight that there are many clever ways by which you could model phenomena which don't tell you exactly what's happening, but it eliminates a lot of possibilities that you are otherwise thinking about. Yeah, so that's all I'm going to talk about as far as modeling goes in this talk, this part of the talk. We'll come to a different type of modeling later. Yeah.
Is there, in your model, is there a space-dependent switching rate?
Yes.
So
There is a space-dependent switching rate. And there has to be because we know this is a spatially constrained system.
And that's like adding by hand, space dependence right?
You mean to see how it expands?
Or like the pattern formation?
No, we didn't have any space-dependent constraint here. It gave this on its own.
Switching rate is not space dependent?
The switching rate is not space-dependent. We didn't impose any space constraint in this model. We have another model now, which has now finally, because we know so much about it, where we put this space dependency. This one didn't even require that. It started producing this all on its own. So that's why I thought it was a really nice model. We didn't bias this. In fact, we didn't really need the model for the paper as well. We just did it on the side, and it showed all these things. So we thought it was useful. Okay.
Just a quick question. So in the model or the experiment, did you consider any factor of media replenishment per se? So how frequent was the media?
There is no media replenishment. This is a plate. You put the colony,
Okay.
5 days, 6 days later, and it goes on. And what I'll show you a little later is something 20 days later.
Okay. So my question is now, if media was not replenished, the low glucose is considerably low, right? Even at the starting point.
Almost.
So after what point does the low glucose even go to no glucose, and the entire study was?
Yeah, hold the thought. We'll come to that in just a little bit,
Sure.
as we actually really understand. This was just a quick little intro part to today's main.
Sorry, Avino. One last question is, there are a bunch of parameters in your model. Did you determine them from experimental values?
This one had actually very little experimental thing. All Vaibhav did in this model was to say, predominantly, you would use aspartate for carbon to make trihalose and put a minimum score for making nitrogen to make nucleotide. It was not an actually experimentally informed model.
Okay, so if I play with the rates, what would happen to this pattern?
Yeah, we have all of those controls and all of that. You can start seeing changes in the colony morphology, which again, we didn't do exactly fine-tuning rates, but we can say that at some point, if you stop it, you will stop getting expansion of colonies or not, or you'll get too much expansion, but then it collapses. So we had the collapse also happening. It came on its own. Again, the model just gave those results without imposing any of those constraints.
So
we can talk about that a little later.
Quick question. Yeah. Remind me what trihalose is and does.
Trihalose is a gluconeogenic product, which is 2 molecules of glucose. And if it's consumed by some cells, it can drive glycolysis. Just write this down.
Yeah.
Okay, so you have G6P, trihalose. Trihalose is actually G plus G. If this is broken down, you will drive glycolysis. If you go backwards this way, it's gluconeogenesis.
Is it like a smaller version of glycogen?
Well, glycogen is a little different. It's also from here. That is a long poly chain of glucose molecules. You can break down glycogen to give glucose also. So these two actually are made together coincidentally, but cells, at least yeast cells and so on, prefer to use trihalose for a variety of reasons. It's more efficient when you break down to give you that burst in glycolysis. But glycogen kind of does the equivalent in mammalian cells.
So in your model, I want to know the parameters here. You're just controlling how much of that source
if
is
we
using
have aspartate. Wait, let me show you this here. Aspartate can primarily be used for making this, or be primarily used to help make more nucleotides.
So you're saying a fraction of it gets used up to make the to run gluconeogenesis, and a fraction of it goes towards nucleotide synthesis. That's the only control.
It's a relative balance.
The fraction that goes there, the ratio of the fractions.
Yeah.
But that how is the spatial?
Hold off. Just it'll come. We'll come to that. We'll exactly show you the spatial part. There's a mic there.
Just a question about the experimental numbers. So this switching that you're talking about from one type to the other, I presume it takes some time.
5 days. Don't get any light cells for about 4 to 5 days.
And is it symmetric or is it asymmetric in the sense of switching from one to the other? Is that the same amount of time it takes from switching?
We don't know that. This is a very nice question. We've thought about it for a long time. We don't know exactly, because in this system, we don't know how to follow a single cell and observe and see the switching rate. So we don't have this switching rate information.
The reason that I'm asking
in
is,
this model, though, he had assumed Vaibhav had assumed symmetric equal probability of switching based on how much the resources.
I see. Okay, so that's very different from what I know from E. coli, because in E. coli it seems like glycolysis and gluconeogenesis show what's called a growth lag trade-off.
Both have a growth lag trait. So yeast have a growth lag trait. We know that experimentally in liquid culture, where we can measure this and follow it. But in the colony, we don't know if the relative rates of switching are particularly different. So,
Okay.
so I'm just saying, yeah, it's likely that there is a lag. The switch from glycolysis to gluconeogenesis should follow a different rate from gluconeogenic to glycolytic.
Exactly.
In liquid culture, it does. We don't know how much is happening in the colony. What we are seeing is a lot of
Because it's
different
hard to track experimentally. Is that
very hard to track.
Is that why? Okay.
Very hard to track.
So
We
this
can't-
5-day number, where does that come from?
we observe the colony developing, right? And we have bulk reporter information. So at a bulk level, we practically don't see any glycolytic cells in the colony for about 4 days to 5 days, somewhere around then. And when it comes up, so gluconeogenic cells, that was kind of go up like this and then they plateau. Glycolytic cells, you're like this and then it goes up. This is a very sharp, this is around 4 days or so. Yeah?
Okay.
And this then they go up very fast. In one day, you go from almost 0 to 20% of the population.
Thank you.
Yeah. Okay. So this is the experimental part where we could actually try to see what was happening relatively in the light or dark cells. What we fed was 13C aspartate and 15N aspartate. And here we can measure the relative flux to trehalose. You can see that in the dark cells, all the 13C aspartate is going all the way to trehalose. So this demonstrates that this is happening. Aspartate is being converted here and going all the way here. I'm just showing you this end, but we measured everything along the way. Okay. Now, relative to the light cells, this is about 4-fold higher or something like that. You can see the reverse experiment of aspartate going to nucleotides, and we're following this nitrogen here. And you see the exact opposite result in the light cells, where you see the nitrogen now from aspartate, relatively more of it going to nucleotides in these glycolytic cells. You can see that there's essentially a budgeting of the same resource that's different in these two cell types. Okay? So what we're really seeing is a division of labor that's coming from a division of biochemical or metabolic labor. Of course, at the level of the organism, and I'm only including this bit because somebody asked me a question on Wednesday. That's not really the main focus of the talk. But if this was actually useful for the community, then there would be some use of having the light cells and the dark cells growing together. Otherwise, why not just have gluconeogenic dark cells, right? In theory, they should be fine. They're all doing gluconeogenesis and doing okay. So we experimentally looked at that to see if there was finally a division of labor resulting in different properties of the cells collectively helping the organism. What we find is this. So the gluconeogenic cells are extremely good at surviving. So they survive freezing and thawing or desiccation or heat stress or whatever it is relative to the light cells. The way we do this is we take the whole colony and then we subject them to this, and then we isolate the light or the dark cells and then see what their viabilities are. And you can see relatively the dark cells have fantastic viability, much higher survival and so on. But collectively now, it looks like having these two states, because their now entire physicochemical properties are different, gives the whole organism, not organism, whole colony, I'm sorry, I should not use the word organism, whole colony, tremendous advantages. So for example, what we see over a long period of time, this is now 20 days or so on, the cells without gluconeogenesis are struggling to grow. They kind of just grow badly. The ones which have now gluconeogenic cells, but they cannot consume trehalose, they grow quite well, but they don't expand nearly as well as the wild type collective colony. So these are able to grow much larger and bigger over time while having the two states. Separately, you can also see clever things like this. Now, if you provide a glucose source far away from the colony, the gluconeogenesis mutant or the mutant that cannot break down trehalose cannot forage and migrate towards a glucose source. This collective colony can now nicely migrate towards a distant glucose. So what you're seeing is that deep down with this budgeting of this one resource, you get two state populations, which collectively now allows you, the entire community, which is a clonal community, to have adaptive advantages in growth, in survival, in proliferation, and so on and so forth. Okay, so this was a follow-up study. This was also just the introduction to today's talk. It's so good we wasted 20 minutes on it. Very well, thank you. Yeah. Yes. No, there was no rescue experiment per se. You mean in the colony? Yes, yes, yes. Yes, yes, yes. You get a very nice, you get all of these phenomenon recapitulated. Quite well within limits of our experiments. But you see a very nice rescue of collective community survival, success, foraging, all of it. Yeah, so when the reconstituted experiments, we don't have any aspartate in it. We do it with minimal reconstituted. So the colonies, the wild type colonies are in complex media, but when we do reconstitution experiments, we make complete minimal media and then add back everything in individual components. So we can see aspartate providing a substantial fraction of the rescue. Yeah, so we've done it both ways. I mean, it's a lot of work that's gone into this. Okay, so
sorry, can I ask you? So you mentioned that that the colony is clonal, right? And it seems important because evolutionarily you don't want to support invaders. So I wondered whether you have whether if you've looked into or any information about is there signaling, like do the cells that produce trehalose try to make sure that the ones that will take it up are actually their siblings?
This is a very, very good question. A really good question. Actually, I don't think clonality is really important in this context, except that it is for yeast. But in theory now, if you look at something like this, if you had somebody else that was consuming trehalose well, it could do well to eat up the trehalose that this colony is producing. And that would change the dynamics of this system. So we are actually now doing synthetic experiments by making different types of mutants and mixing them together. I'm just saying all of this in a long-winded way because I don't have a definitive answer to your question. But the idea is exactly this. If it's clonal, there should be some advantages of doing it for a clonal community. Of course, we see it here, but there's no one else over here. Now we're starting to put in different combinations of cells which can either consume or produce trehalose at different rates to answer this exact question. I don't have the answer, so I don't want to hand wave and give you a wrong answer.
Do you have some understanding why it's better to have this division of labor across different?
That's the whole rest of the talk, okay? So, and it comes back to the original question we are asking, which is what is the need to have different metabolisms going on? So that is the rest of the talk. And the rest of the talk, this is unpublished. This is really exciting new work. Essentially, what we're saying are what are the biochemical constraints driving spatial specialization and division of labor. In this case, I'm just going to focus on division of metabolic labor because what we have is this separation. One is highly glycolytic, the other one is highly gluconeogenic. It's a division of metabolic labor, right? This is, of course, a clonal community. In theory, though, I don't see any reason why this can't happen with, say, two different species, one which is very good at one and one very good at the other. For that question, we don't have the answer. Okay. We're asking what is the organization of this whole metabolism? And I'll also introduce some of the experimental approaches we have used here, along with the computational approaches we've used here that we've tried to integrate. Okay. So, of course, at this level, all I've told you about are just these two pathways, glycolysis, gluconeogenesis, and our approach was very reductionist. We built it up from like starting point. We didn't know what was happening at the level of the whole cell or the whole genome. So now because we understand the system so well, we're asking this question at the whole genome scale. What we do now is this is the colony. We can be micro-isolated. Sriram did all of this. When I say we, it's all some individual in the lab. In this case, Sriram, who was lead author on the first two stories, along with a couple of other people, Piyush, who did the modeling in this part of the story, and then Adish, who did a lot of the metabolism, and Rajalakshmi. Okay, so we micro-isolate. This is a colony. It's about a centimeter, less actually, and under the microscope, you can isolate these light or dark cells carefully with a little needle or tweezer. We collect these cells, and now we can do RNA-seq analysis of all these cells to look at the global gene expression profile, right? What we of course saw, which was very satisfying, is that there's like a nice clear separation of these gluconeogenic state cells and glycolytic state cells. They beautifully separate into entire genome scale transcriptional profiles that are so distinct. That was beautiful to see. But when we started organizing now the types of gene expression changes that you see something beautiful at the whole level of the entire metabolic network, at the level of the whole transcript. Okay. So for example, the light cells or glycolytic cells are showing you lots of all these kinds of things. Ribosomal biogenesis, glycolysis, of course, these are grouped together because most of the enzymes are the same, but I'll tell you what's different. Amino acid biosynthesis, tRNA biosynthesis, all of this is looking at other types of pathways, fatty acid breakdown, glyoxylate cycle, gluconeogenesis, TCA cycle, all of these are very, very high over here. What does this mean? Actually, if you look at it just at the level of gene expression signatures, you see this beautiful separation. Light cells, for example, have very, very high nucleotide biosynthesis and amino acid biosynthesis. Light cells also have very, very high ribosomal biogenesis. When you see this, of course, when you see this at a systems level, the first thing you think of is this kind of a signature is a reflection of a growth state. It's a perfect reflection of a growth state. Take a cancer cell, it'll also show you the same thing, right? But there's something more. What we wanted to now see was what was the deeper chemical logic interlinking each of these to see if then the chemical logic will explain these two states. And we had a beautiful data set, which is a very different system. I'm only introducing it in part because Jonathan also talked about biochemical oscillators in his talk, in his second talk or something like that. There's a beautiful oscillatory system in yeast, one of the earliest observed oscillation, which is a purely metabolic oscillation. This is called the yeast metabolic cycle. This is in well-mixed liquid cultures, so there's no spatial separation. They're just constantly spinning. This is a chemostat. You reduce glucose to a very, very small trickle, and the cells spontaneously start oscillating in oxygen consumption. Okay. These are long, slow oscillations. Each oscillation takes about 4 hours. Essentially, what you see here is this increase in oxygen consumption of the entire population. When we looked some years ago, looked at temporally what's happening to gene expression all over here at each of these points, they actually grouped into 3 very distinct biochemical states. Okay. This state, this red, where you have this high oxygen consumption, highest oxygen consumption, correlated with this growth state type of a thing. You had lots of ribosomal biogenesis, lots of glycolysis, stuff like that. Cell division was nicely separated to this state, and this state had what Ben had originally called some kind of a survival quiescent state. Okay. And we have very fine resolved data sets of the entire transcriptome of each of these states. What we did was compare what we saw in the light and dark cells to the gene expression profiles in each of these to see if it correlated with each other. Okay. And so this is the data. Where is that? Here it is. Each of these spots is a gene expression profile here where we correlate our gene expression signature with this. You see 2 beautiful things emerge. The dark cells perfectly or very highly correlate with the gene expression signature of this state just before they enter growth, while the light cells almost perfectly correlate with this ox state. Essentially, what you see is that the light cells have a perfect expression or a correlation of a growth state, while this one is a late preparing for growth state, 2 perfect separations. And this state is highly oxidative collectively, and this is a reductive charging state. That's how it was defined in the yeast metabolic cycle. Is this clear? I'll just summarize this over here. Essentially, what you have collectively is an overall reductive coupled state in the dark state. There's a lot of amino acid breakdown, maybe autophagy, gluconeogenesis. You're making storage sugars. This is reductive charging. And the growth state is an oxidative state where you have glucose catabolism, cell cycle, amino acid, all of that.
A quick question, if I may.
Vijay.
Previous slide. Is that curve of oxygen consumption a population level measurement?
Yes.
Or
So this
single
is
cell?
large bulk population. This is in fact a very dense culture.
Are there similar single cell measurements?
There haven't been so far because every time you take the cells out of the chemostat, the oxygen oscillations collapse. I did that 10 years ago. I tried very hard with a microfluidic system. We never got very far, but it's worth revisiting this now that we can do all the single cell tracking much better. So we don't have the answer, but it's something I'm thinking of revisiting. Amino acid breakdown, gluconeogenesis, some autophagy, preparing lipid molecules. You're basically carrying out a whole bunch of anabolic and catabolic reactions to build up the molecules you'll need so that you can shift to a glycolytic state. The glycolytic state is really needed to sustain ribosome biogenesis and nucleotide synthesis and so on. So they're actually, each of them are coupled. You need to do one to the other. In fact, many people think a cell itself will go through this internal cycle of build up and breakdown, but that we don't know. But at least at a population level of cells, you see this separation now. The difference though is this was temporally separated in a mixed population. What we're seeing is this spatially separated in a fixed population. They can't move, right? So we see this. Okay. So the highlight of all of this comes back to what I've been talking about. Key nodes in major pathways are essentially mutually incompatible. So if you look at glycolysis this way and gluconeogenesis this way, you have these key steps here where you cannot easily reverse in the same using the same enzyme or the same reaction. So the PFK step usually favors glycolysis. This delta G is -17. You want to go back, you use a different enzyme and use a different cofactor to get back here, which is FPP1. Here PP goes to pyruvate this way, huge delta G barrier. If you want to go back, you go back this way to create a slightly more favorable delta G regime. The net of this is negative, right? And what we see in this gene expression signature is for most of these things, gene expression is not different, not too different, but the key enzymes here that would be gatekeepers are completely contrasting in light and dark cells. So PFK and FPP have opposite expressions in the light cells and the dark cells. And then PYK, which is this, completely opposite expressions in light and dark state, which means they have perfectly trapped or opposed their metabolism to carry out opposite reactions. You can also look at this in terms of feasibility of the overall thermodynamic feasibilities. These are calculated based on rough numbers that we have. And you see that in the dark cell, your gap TH reaction, which is here, is in an infeasible regime based on what you have inside there. If you want to bring it down to this feasible regime, you have to shift to this light cell regime. Okay? So at this point now, I've left you with this experimental evidence that the two populations of cells have completely separated their overall genome level gene expression signatures based on the separation of their metabolic processes. The idea then is that the metabolic changes drive the overall gene expression changes, but I'm not going into that too much today. I'll stick to the metabolism component. Yeah, of course. Yeah, we have that data. The gluconeogenic cells grow about 30 to 40% slower than the glycolytic cells. So if the doubling time of a glycolytic cell roughly is about 85 minutes, the doubling time of the gluconeogenic cell is about 110 or so minutes. So we know that difference is there. And that also eventually explains the expansion of the colonies that we see. So the glycolytic cells keep dividing faster and expanding, and then they switch back and all kinds of things happen. Yeah, so that's what we are saying. I hope it's true. Yeah. The causality bit is a little trickier, but I'll come to that in a bit. See, so good question, very good question. We could not measure the exact growth rate in the colony because we can't track the single cells as well. What we do is we micro-isolate a light and a dark cell, shift it back to either, so take that same cell and shift it to a same exact medium gluconeogenic, or shift it to a high glucose medium and look at the initial growth rate. For those liquid cells when we transfer. So there is a fudge over here. Liquid. Yeah, but cells taken from the colony. So that's the only way we could do it now. There is a fudge here. It is not the colony exact growth. Okay. Colony, we have only gross bulk measurements, overall dimensions of the colony, and relative populations of cells at any given point of time in the colony over days. We can't find dissect. Another question.
I was just wondering, what you're proposing is that, you know, under glucose or low glucose conditions, you get the temporal separation between, you know, the reductive charging, the classical metabolic cycle, and now what you're proposing, you have a spatial separation of these two states. So, and that comes a little bit back to the question of the single cells. Is the temporal separation in the yeast metabolic cycles, are these the total population with shift in time, or is it a subset of population which are growing, and you also have the spatial separation?
Yeah, exactly. So this
and then second question is, if you watch the colony growing from a single yeast cell, when does the spatial separation arise? And how are they shifting states if you follow a trajectory or a lineage of a single cell? Or is it always separated so they are not be able to
Exactly.
shift
So this
states?
is an absolutely great question. We have discussed this endlessly, exactly these two questions. Okay, the first part, in the yeast metabolic cycle or whatever, it's still unresolved whether it's the same subpopulation of cells dividing or not. Now, I think, and we've theoretically proposed it, that it is not, because now looking at or understanding this, if it's driven by the resource balance, if in a certain cycle, a bunch of cells divide, they will drop in their internal resources to lower, while the others are better prepared to make the next shift. So in the yeast metabolic cycle, only about 35% of the population of cells divide in each cycle. It's not 100%. It's not a complete doubling. We know this information, and we tried to track this at single cells. We couldn't do it because once you take it away from the chemostat and the mixing, you lose the oscillation. The oscillation dies within 1 or 2 cycles. So we don't have an exact answer, but we've proposed or believe that it is a different population of cells in each cycle. Basically, if you go to the resource threshold model, if you need so much of the threshold to switch, whichever cells are closest to this will make it in the next cycle across and will switch, while other cells that are trapped in this regime are too far away from this threshold to be able to switch. Second question, in the colony, following the same cells in real time, that's exactly what we want to do. We haven't found an experimental way to successfully do that yet. We have tried really hard, like really hard. Part of it is just the challenge of imaging colonies itself. Very hard to do at the level of cells, small cells all piled one on top of each other. So we haven't been able to do this because all of these are high population dependent phenomena, right? We only see the switching after 4 or 5 days. There are millions of cells in the colony, so it becomes very hard to resolve or follow a cell. We want to do that. We absolutely want to do that.
I have a question. So in a colony of yeast cells, when you see a few cells are like there's a distinction between light and dark. So there's a distinction between either gluconeogenic cells or glycolytic cells. Do you observe any hybrid phenotype?
Yeah, exactly. I mean, that's the thing, right? We should. So what we did here, I'll just tell you what we did here. That's a great question. Absolutely great question. What we did here is isolate where they were very clearly light and dark. We wanted to keep them separate for this reason. There obviously must be, and if you collect the whole colony and sort, if we get something, if you're, now we know the gene expression signature, so I'll give you a distribution. This is number. This is kind of state. Let's say this is gluconeogenic. And this is light and dark. We had isolated this and this, but there are a bunch of cells that are in this regime. They're there. Now, I'm assuming those are the ones that are slightly here, and maybe today or in some days or months or years from now, we'll be able to answer what those intermediate states are. They're there. Okay. But these are the two that are driving the whole population, and I'll show you why, the whole system. Okay. So now the other thing is, the one reaction we are particularly interested in is this. I told you that this seems to drive the whole network. Aspartate being converted to eventually to trehalose, right? So first things first, we have to see whether the relative utilization of aspartate is particularly high in the colony. So we did this rather cool experiment. This is the colony. It's on a plate. We actually looked over time, taking out the media right beneath the colony to see the relative usage of amino acids. Okay, this is which one is being used the most, which one is being used the least, based on measuring what is present in here. And you can very clearly see this is aspartic acid. These are all the other amino acids. They're using all the amino acids at different rates. This is way high. So it's suggestive or supportive evidence that this is really key to the whole system. Okay. So now though, there's a second part problem. You see that this is the minimal equation we're talking about to drive the whole system. You see there's another thing that's needed here, alpha ketoglutarate. What happens here is a transamination. You take an amine from here and put it here, you will get oxaloacetate and glutamate, which means you need a steady supply or source of alpha ketoglutarate. Except that most alpha Kg is in the TCA cycle inside the mitochondria. In fact, made by this isocitrate dehydrogenase to make alpha Kg from isocitrate. This is required for energetics. Now, I told you there are multiple ways to get to an end. It turns out that enzymes sometimes have isoforms or equivalent orthologs that do the same reaction. Yeast and actually most other cells have two versions of isocitrate dehydrogenase. They're both actually mitochondrial. And we hypothesize that maybe these are required to produce different subpools of alpha Kg. Presumably this one, which is considered non-essential, is only useful in a gluconeogenic regime to provide alpha Kg for gluconeogenesis. And the way we would do this is to see what happens when we knock out only this to the production of trehalose, the formation of the colony, and overall energetics. This one you cannot knock out because it's essential for the cell, it's driving the TCA cycle, right? This is one part of it. This is the mitochondria part. Second, you need a steady supply of oxaloacetate. What is the most efficient way to make oxaloacetate? See, the other thing that's going on over here is the TCA cycle continuously, but all this oxaloacetate is inside the mitochondria. Gluconeogenesis, like glycolysis, in a eukaryotic cell is completely cytosolic. This pool of oxaloacetate is not available for gluconeogenesis. That means there has to be another way to make it. Turns out organisms have a wonderfully clever way to do this. It's called the glyoxylate cycle. Initially, it was thought to be half the TCA cycle because what you do is oxaloacetate, citrate, isocitrate, and then you kind of basically jump from here to here. You jump via glyoxylate to malate. You don't do this whole cycle. No alpha Kg, no succinate, no malate and all of that. You just jump over here. This whole process is entirely cytosolic. And what we see nicely in these dark cells, this is relative light to dark. You see very, very high expression of the glyoxylate cycle in the cytoplasm, very low expression in the light cells. So this first part, it shows that the glyoxylate cycle appears to be very important. Yeah. And the second part, which is this alpha Kg business, providing alpha Kg, we have these two isoforms. Now, if we knock out this isoform and we measure just production of trehalose and the formation of the colony, trehalose production drops a lot. The colony no longer gives you light cells, but it doesn't affect basic energetics, which is what is being supported by IDH1. Mitochondrial function, activity, ATP production remains the same. So it clearly shows that this alternate pathway to make alpha Kg is really important to provide a pool of this metabolite to support this additional function. Okay. So now I've just shown you stuff with the basic system, focusing on this one reaction, this aspartate to glutamate. But still there are some problems over here. If you just do a whole balance sheet, we're still not sure how we're getting collectively enough acetyl-CoA, which is kind of at the center of the universe, to drive all these processes forward. We are actually fudging our balance sheet. That equation looks important, but it doesn't seem sufficient to sustain this colony over days now and weeks, actually, 25 days, 30 days. This whole system should collapse. So something else must be happening.
I actually had a question
me.
from the previous slide. So you're adding the aspartate, right?
It's there in the medium. This is complex medium.
Yeah. So the oxaloacetate that you have, that you are already getting from the transamination reaction.
Yeah, using aspartate.
Yeah. So how exactly is the cytosolic cycle becoming relevant?
I'll come to that in just a moment. Just hold that thought. I'll tell you how it becomes really relevant. But you need alpha Kg. What you're presumably doing here is making this pool of alpha Kg separate from this pool. This pool is coming outside. We don't know how exactly that's happening, but it's available for gluconeogenesis. Okay.
Yeah.
It's providing this to give you this.
That I've understood. I was just wondering about the cytosolic glyoxylate cycle.
Yeah. So the glyoxylate cycle, what it does, where is the glyoxylate cycle? Here is by shunting this way, helps you boost the production of oxaloacetate, and this is all in cytosol. So you're basically taking cytosolic citrate, isocitrate, and boosting the production of oxaloacetate, providing more oxaloacetate continuously here to presumably supplement what is going on over here. This is providing you some, this is providing you some more. We will resolve this in 2 slides, okay? We will resolve the balance sheet.
Just quick question. You could also just oxidatively deaminate glutamate, right?
We could.
Via glutamate dehydrogenase.
Yes.
Did you knock it out? See what happened?
We've reduced that. It doesn't affect it as dramatically as this. There is a contribution.
Yeah.
So actually, this is a very good point. When we're talking about contributions, we're talking about relative contributions. And at least at the initial stage, this reaction seems to be very important to drive the reaction forward. Now, as you go over time, I'm still telling you something, it's a snapshot at 5 days or 6 days. We're going to see the proportions of each of these change as the community and the colony continues to grow. So you have a continuous remodeling of the metabolism to help this stage.
No, because they would help you with the alpha
Exactly.
ketoglutarate, right? Like
Exactly.
you make glutamate into alpha ketoglutarate.
Exactly. It should. So we see some of that, but the timelines shift. So the important thing again coming back is how important timelines are in biology, in development, in organization. Okay, so I'll come to this part now. How do you sustain this whole system, right? And this is where we actually find use for bulk genome scale metabolic modeling. What you do in a genome scale metabolic model, I think we introduced this a little bit, and Nitish must have gone over this in a tutorial. You take all the reactions that you know this genome might have, assume them to be all present, and then build a whole metabolic network. And if that's functioning, so this is an unconstrained metabolic model, everything is present. Now, if we separate to light and dark cells, you basically show that you have very high gluconeogenic and TCA in the dark cells, and in the light cells you have high TCA, presumably more glycolysis and so on. So the gluconeogenic part is inactive over here, right? And then what you can do in a genome scale metabolic model, it is a steady state flux balance model. So it's not really telling you everything that's changing in the cell, but it tells you what happens when something is there versus not there. You can use it to optimize for a certain parameter. So in this case, we optimize for consuming aspartic acid, because so far I've told you that that's what's driving the whole system. To make biomass. Biomass is what you need to grow. That's your measure of cell growth or so on. Now, when we do it this way, this is what the theory tells us. Yes, you can make lots of Trelos with aspartic acid, okay? Except when you do that, there's a massive trade-off between growth and production of the resource. If you want to make high amounts of aspartic acid, over here, you can make lots of Trelos, but you have a massive trade-off with growth. Every time you make more Trelos, your growth rate drops very dramatically. Now, collectively, though, we don't see that happening in the colony. They're growing quite okay. They're not growing that badly, which means, at least according to this model, our experiments, our conclusions, there's a massive trade-off between growth and biomass, which cannot be explained with what we have. Right? So is that clear? So that means, can aspartate enough be sufficient to sustain the whole system? That really becomes the question. Or is it really important at a certain time regime and later is supported or supplemented by something else, which comes to how different pathways can come and help you do your job, right? And of course, experimentally, I showed you this business of how important the glyoxylate shunt is. So now, if we put back the glyoxylate shunt in this genome scale model, give it full activity or whatever, you can see what happens. And what we do there, this is theoretical, we can do a flux sampling, look at isocitrate lyase flux, that's the overall isocitrate lyate flux, sampling across different possible substrates or carbon sources or whatever, different amino acids and so on, and look to see where you can relieve this bottleneck of growth versus biomass production versus Trelos production if you use only aspartate as your sole carbon source. And the only way you relieve it is when a new molecule comes into the picture, which is ethanol over here. You can completely relieve this biomass resource production trade-off. So you understood what I said? Was that clear? Did I go through it too fast? It's the one place where I found people get stuck. Huh? Didn't understand. Okay, good. Thank you. Anybody else? I'm sure there are at least 5 other people. So we will go over this again. So what we have here is if we have only aspartate in the model driving Trehalose production, you can make a lot of Trehalose, but when you do that and you plot it against potential growth there's a massive growth trade-off. To make a lot of triallose, you have to give up your ability to make growth. Oh, it's not mine. Here's mine. Dark cells. Aspartate is the gluconeogenic substrate. You require that to sustain gluconeogenesis to make triallose. The triallose is being consumed by the light cells to drive glycolysis. That's the two-state system. Just be clear on that. In the light cells, whatever aspartate is there is being consumed by the light cells to make nucleotides. But that is not in conflict with any of these things. There you need only some amount of aspartate, not a whole lot. It is helping make more nucleotides, which is a feature of a glycolytic. Okay, so here this is the conflict that happens. If you make more triallose, you will do it very well with this pathway going this way. But then when you do that, the cells aren't able to make biomass. This is at the level of the whole network. We don't even worry about what all are happening in the network that are not sustained. The genome scale model tells you this. It can't make this much biomass or sustain such a growth rate.
So, but this is in dark cells.
This is in dark cells.
So could it be possible that colony growth is driven by the light cells?
Yeah. So we'll come to that. We'll come to that. So how do we resolve this trade-off? That's the whole point. We still see expansion of the dark cells. It's not like they're not growing. They're growing, they're dividing, the colony is expanding. We have 70% of the cells being dark. So that means you can't have this kind of a resource bottleneck. What we know experimentally though is we need this much amount of triallose to sustain the system, which means the math isn't adding up. You're fudging the numbers, right? How do we see what is happening? What we do now is look at triallose production, but now start providing all these other things into the whole network to see which will help drive the glyoxylate cycle flux towards and other pathways. We reintroduce the glyoxylate cycle, we bring in other things. So all these things come into play to see which channel to triallose. And we see that the only way we can resolve this bottleneck is when you have a huge excess of ethanol in the system. Okay, this then completely relieves this bottleneck. I'll just show it to you in, yeah, here. Basically what it's saying is now if you're having aspartate production, if you now keep increasing ethanol uptake, you start getting lots of triallose production without a trade-off in growth. The growth then gets restored to high levels of growth. You're not wasting your energy trying to make triallose from aspartic acid, and it balances out. Okay, this just tells you that at this stage. So essentially the idea is this. Maybe what's happening in the system is initiating the system. You have aspartic acid, and these cells are making triallose slowly but steadily and growing slowly. But at some point, you build up enough of this, and you start switching to light cells. Now, what can a glycolytic cell do? This is the original idea of what we talked about on how glycolysis is organized. When you do glycolysis very fast, you ferment, and you make ethanol, which means once these cells emerge, they can start putting out a new resource, ethanol. This then can be consumed by this. This will massively boost this one's efficiency to make triallose while allowing it to continue to grow. And so you now have from a low efficiency system to a high efficiency system. And this is enabled by the production of this new resource in these wasteful glycolytic cells. So you have this thing called overflow. They're overflowing, they're wasting ethanol. This is helping the system. So essentially what you are hypothesizing is a perfect two-state mutualism to enable each cell to maintain its highest metabolic efficiency. Okay. So now ethanol becomes a substrate for these cells, and they will use this to no longer rely on this to make triallose and use this to sustain the production of triallose over time. No, everything else remains the same. The dark cells remain the same, and I'll tell you why in the next slide. Okay. They don't have to do anything. They can just start utilizing this, and they become even more gluconeogenic. They just continue to reinforce their existing state to an even further gluconeogenic state and drive the production of triallose. So experimentally now we have to see if this really happens. Okay, so what I've essentially said is you can create a perfect two-state mutualism. One state cannot exist without the other state. The gluconeogenic state is essential for the glycolytic cells to even emerge. But once they do, they provide something that feeds back here to maximize the metabolic efficiencies of each of these states. So how do we test this? First things first, if you want to consume ethanol or make ethanol, if you're glycolytic, you have this enzyme ADH1, which converts pyruvate to acetaldehyde to ethanol. And if you want to consume ethanol, you use ADH2, which is the reverse direction. You break it back and you move it back towards gluconeogenesis. The gluconeogenic cell should have a lot of this and less of this. We see exactly that. You see completely opposite expressions of these two enzymes or isoforms in the light and dark cells. They're perfectly opposite in just this one endpoint resource, right? The next thing now is what happens to the whole network. You can look and see at just this cross-feeding system. In the dark cells, you have massively high expression of this reverse reaction that will feed into producing acetyl-CoA to make trehalose. And in the light cells, you have a perfect opposite. It's very high for all these glycolytic enzymes, but nothing for these reverse and nothing for this. Okay. So now the key experiment is you only block this step in a mature colony. What should happen is that trehalose level should drop, the colony should break apart, and that you won't have light cells anymore. Which means just blocking this should collapse the whole system. Exactly what we see. This is a knockout of ADH2. Now you still have highly gluconeogenic cells. In fact, gene expression wise, they remain very highly gluconeogenic, but they can't make as much trehalose anymore. And there are no longer any light cells in the system, which means the entire system collapses. Okay. And now you can also model this back to look at overall aspartate flux, ethanol uptake flux, and overall efficiency of the system. And you see exactly this. If you now continuously provide ethanol uptake, you're at the maximum efficiency regime. If you block that, your whole two-state system will collapse completely. So here really is the idea that we're putting together. We have gluconeogenic cells. They're making a resource slowly but steadily, right? They're taking an aspartic acid or something like that. In this case, it's aspartic acid. Low efficiency, slow buildup of this. But if this builds up to a high enough state, some cells will switch to this regime. As they do this, they shift to this completely opposite metabolic state, oxidative state. Their whole gene expression signature is presumably a consequence of switching to this state, and they produce another new resource. This new resource is not of great use to this, but it's tremendously useful in relieving these metabolic resource production versus biomass conflicts, so that you collectively have a system that functions together to continue to expand over time to make all these other precursors that you need at the base. So essentially the bigger idea now, we come back to this question earlier of whether it has to be same species or different species or so on. You're basically thinking of purely biochemical ways to create a heterogeneous population of cells with interplay between each of them. So this is the idea then. Supposing you have a cell, it's using some resource, whatever is there in the environment. It starts making new things that a new cell can switch to use. It could switch to growth state or whatever other state. But as it does this, it starts making a new resource which would feed back here. And presumably then you have a perfect mutualism that enhances each of them to be what they are. Now here I've just said two different colors or something like that. But in this case, the cell need not be isogenic. It need not be of the same genotype. It could just be that there's some other cell that comes that's very good at utilizing this resource over here. If you're looking at a community of cells, of microbes, you see this all the time because you do see existence, very common existences of auxotrophies of key metabolic pathways, not just amino acids or even sometimes central metabolic intermediates are missing. It could just be that the two, one way could be that the two are actually mutually enabling each of them to function better. Of course, there are many other ways. You would have cheaters, you would have excess resources produced. We're not getting into that. Focusing on this idea of metabolic efficiencies, this is just an idea. So little take home though, what we see is that by spatially separating processes, you make mutually impossible biochemistry possible. These two reactions would not otherwise be going on at the same time and same place. You can either solve it temporally, like the yeast metabolic cycle, or you solve it spatially by putting them in two different places. And then there is actually real use of things like overflow. Overflow was this excess glycolysis producing ethanol. You're actually producing new resources that somebody else can use. In theory, it's not very different from, say, what many cells in mammalian tissues are using. Some cells produce a lot of lactate. Other cells take up lactate and utilize them. This we know very often around tumor environments, around macrophage environments. And what this allows, this last thing, I've particularly drawn it like this because this is metabolic closure. Starting with something and you're not leaving things open-ended, you're basically tying in loose ends by completing the whole metabolic box. And you're basically allowing metabolic closure to help solve the resource allocation problems, except that this is at the level of many cells and not just one cell. So maybe this is a way to get to cellular complexity, or maybe the simplest way to get to cellular complexity. Okay, and I'll leave you with this slide because this is a really famous example. The most dramatic visual example of complete separation of metabolic processes is the reason why we are all alive. It's photosynthesis and nitrogen reduction. Plants don't do photosynthesis and nitrogen reduction at the same place and at the same time. Photosynthesis is spatially separated and temporally separated. So in this case, both, because there's a day and night. Photosynthesis is highly oxidative. Nitrogen fixation is a highly reductive process. They're completely oppositely separated in the leaf and in the root, and in the day and at the night. So you actually see, this is of course very famous textbook example, but we see this kind of ability to help close all these metabolic reactions, even at something as simple as a colony of yeast. Okay, I just want to acknowledge all the people who were part of this work. Sriram started this project. Lots and lots of people have chipped in at various levels. Vaibhav built the first coarse grain as well as the agent-based model. It was a beautiful model done with Vaibhav and Sandeep. Adish drove all the analysis of the metabolites and the metabolic flux. Piyush built the genome scale model and actually identified this resource versus biomass conflict. And that really helped piece things together in the story. So thank you very much. And I will have some more last questions before we wrap up. I've left enough time now, intentionally left enough time for now some of these kind of questions on how you complete a biochemical loop and how you could increase or decrease efficiencies and so on. So I would like just some open discussions on what you think on how you would go about doing some of this.
So can I start?
Yeah.
Yeah, thank you for such a beautiful talk. I really enjoyed it. But that question still remains, like why a single cell couldn't do both, especially in yeast, if it's possible to do the oscillation and doing it temporarily. Suppose all cells would have done Trehalose excretion and then uptake, and then this kind of oscillation could have been also a possible.
So absolutely right, right? It's a really good question. And we actually see these oscillations in liquid culture. That's what they're doing there. So why are they not doing it spatially?
I thought it may be because the peripheral cells have more glucose availability or oxygen availability, something like that.
So first thing, at the time scales of these colonies, after 6 to 8 days, we literally can't measure any glucose around there. Now you're still right. They could still, the peripheral cells could have slightly more. I mean, you might be at 0.001% versus 0.0001%. We don't know, but experimentally we can't see that. The second, which is things that I have not introduced in the system, are things like oxygen, which we know are really important. And second, mechanical constraints or physical constraints. In the periphery, your one side is completely open. And maybe there is a mechanical component to controlling rates of glycolysis. Now, this is a really interesting idea because in several other systems, it's been observed that if you restrain cells, the glycolytic rates change quite dramatically. There's no cause consequence over here. It's not clear. Cause consequence is not clear. But the observation is very, very reproducible across almost all cell types. Yeah. So now there are possible hypotheses, maybe, and it's not at the level of glucose uptake. So the uptake and the transporters are not affected, and you've normalized for amount of glucose in the system. It's there. It's not at the level of uptake. It is at the rates of glycolysis. Possibilities are the hexokinase itself is mechanosensing. I mean, mechanosensing, people love to throw out ideas like this. Not enough evidence for any of that. The other is that some of the glycolytic enzymes and pathways itself are constrained in the cell if your cytosol becomes more constrained or something like that. This is a really good idea. I don't know the answer to that. Okay, so that's one possibility. The second one is oxygen. Now, all these cells are respiring. In this case, even the glycolytic cells actually are respiring in this case. It's not very low respiration. It's actually high respiration. They do have much more oxygen in the periphery. We haven't yet found an experimental way to squeeze and modulate oxygen unless we use some oxygen scavenger complicated experiments. And at the level of spatially with these colonies, we don't know how to do the experiment. It's possible. We haven't imposed something like that.
But actually, it's not going the oxidative pathway, it's going the fermentative pathway.
It is fermenting a lot, but it is still carrying out respiration. It is still doing respiration. It is doing more respiration than a liquid culture, low density yeast culture in high glucose. That we know.
Thank you.
Don't know how to turn it. It's just silence.
I have a question. So, this side. Yeah. So I was curious to know that where does the equilibrium state come between the light cells releasing ethanol, that being taken up by the dark cells, giving rise to trehalose, and that again being taken up by the. So I couldn't find like where a particular population will suddenly start dominating.
That's an absolutely brilliant question. We also don't know the answer to that. We've actually discussed this endlessly to see how we could think of ways, either experimentally or theoretically, to understand this equilibrium. We have actually, we don't have any ideas that are what I think are plausible. I don't know. In fact, if anybody has ideas here, I would love to receive them and see if we can design experiments or something to test that. What we do know is this, though. What we do know is if we reduce the rates of trehalose production, you only delay the formation of the two-state colony, because eventually you will make enough trehalose and you will form the colony. And there the rough equilibrium of about 70% dark cells and 30% light cells is maintained. So that we know. Second, we know that, of course, like in this example, if we block the uptake of ethanol now, we will no longer sustain the system, it will collapse. Yeah. So those are the two things we do know. We don't know where exactly the chemical or the efficient. Part of that is because we don't know how much actually each cell or each group of cells is doing. But at this stage, we don't have a way to experimentally obtain that information.
Right. So
if we did, we would.
Okay. So was the gluconeogenic cells seen in greater amount, if I'm not wrong, like without any perturbation, without the ethanol quenching or the trehalose quenching, the gluconeogenic, so experimentally there are observations, but the explanation via model is still
very insufficient. Very insufficient.
That's pretty interesting.
The other thing we see is, for example, when we block ethanol uptake, we still have a colony, we still have gluconeogenic cells in terms of gene expression signature. It's just that they're not able to make that much trehalose. So gene expression is saying they should be highly gluconeogenic, but experimentally they're actually making less gluconeogenic output because they don't have enough resources to sustain. So that's saying how important mass action and availability of substrates is, even more so than the gene expression itself.
Right. So my second question that I have is, I'm still stuck at the low glucose and the no glucose point. So when the media that was used for growth it has all aspartate and all other components with low glucose, but after how long does the glucose completely get depleted, and the entire observation is actually because there was no glucose in the medium?
No, there is not. There must be some. So what we do experimentally, this is what we do. This is a plate, right? You seed a colony somewhere here, right? Now this plate has 0.01% glucose or something like that. It's all over here. As the colony develops here, we can measure glucose in this vicinity. At this point, after 3 or 4 days, based on our experimental assay, we can't really detect glucose. But still, there is some amount of glucose diffusing from, say, from far away, right? It is probably instantaneously being taken up by the cells, which means the cells are taking up glucose, but we don't actually measure glucose in the immediate vicinity over there. And we don't know at this point of time, over longer time scales, how much. I mean, in theory, I guess we can calculate it. We have some diffusion rate and we know the total amount of net glucose here. Little complicated. At least I haven't found a student who's been willing to grapple with this much. Okay.
Right. So no, why I was interested in asking, because it's one of the same experiments I did with cancer cells, and I saw that the glucose deplete to 0, like it's below the detection limit of the device in 2 to 3 days. So it's in the entire medium itself. So cancer cells are attached to the bottom, right?
That's
so
right.
if I don't know how whether I can go to the perfect location where the cell is attached and see the boundary, but what I used to do is collect the media from the entire dish. So and glucose is 0. I mean, it's 0 mg per ml.
Yeah, we can't measure it.
Yeah,
Right.
it's below the detection limit. So it might be possible that not the presence of glucose or glucose per se, but it is absolutely no glucose which is helping the cells to switch to a gluconeogenic feature as
Yeah.
compared to
Yeah.
the
So
glycolytic.
philosophically, I agree with you. It's very much a possibility. It's just that we don't know how we can actually test this. But I have absolutely no problem with this.
Right. So if a
idea.
control can be kept for without any glucose, everything remains the same, but the same culture
Yeah,
was grown in
but-
absolutely no
But
glucose.
what happens? So there we did that experiment where we grew cells in completely no glucose medium, right? There actually something interesting is happening. When absolutely no glucose is in the system, we of course have gluconeogenic cells. They remain highly gluconeogenic though, and we never see the emergence of light cells. They're just highly gluconeogenic and slowly expanding, very slowly. So there is something there that glucose is signaling, and we don't know how. So it's a really advanced, complex question. As of now, we're not
That's very
touching that.
interesting,
We're not
yeah.
touching that, but we see this and we have seen this, and there's lots. We have lots and lots of repeat colonies of this.
Probably because of the ethanol.
Yeah, but what happens is in a gluconeogenic state, there's lots of ethanol present in the medium, and that is reinforcing the gluconeogenic state, which we see over here. So that's our simpler explanation right now. We don't know if it's right though. It's a simpler
That's very
explanation. We are
interesting.
going with that for now. We'll be wrong.
It's very interesting. Thank you.
Just another question. So when the light cells begin to proliferate, do they grow sort of in waves, or is it a steady state? Is it
Very
constant?
good question. Really good question. Somebody else also asked, why can't we have, you asked this, why can't you have an oscillation of that?
Yeah.
At our resolution, we haven't seen the waves here. But I am actually very tempted. I actually like both of this question and your question. I'm very tempted to think there must be an oscillation in here. We haven't found it yet, but I would love to look for this. We're trying to see how we could look for oscillatory plus minus plus minus.
And if there was an oscillation, would it be something to do with like a buildup of ethanol and
Yeah.
then a release?
Yeah,
of
so
ethanol?
that's what I would hope. If it is a resource-driven oscillator, you have two resources: buildup of trialose, consumption of trialose, buildup of ethanol, consumption of ethanol, and you can model this kind of an oscillator very nicely. There are nice ways to model resource-dependent oscillators. In fact, Sandeep is very good at that, doing two-state oscillators, and it can be modeled, and we can get roughly at very crude, back of the envelope kind of calculations. With that kind of an oscillation, you can still get a two-state system like this. So we've done that. Sandeep's done that. We haven't seen it, but I'm very tempted to look for that as an additional, because the possibilities are all there. Yeast oscillate really well. This would work in forming a two-state oscillator, two-state oscillator. So yeah, but colony is growing. We don't have the resolution now to see the oscillation of the resource. We don't have ways to do that. Roughly, it expands that. I mean, this is back of the envelope. This is all on paper pen, not on the model. But Sandeep thinks it'll be close. You can get it both ways. Roughly same time scales, because these are slow time scales. Colony growth is over days, weeks, right? So in this time scale, it'll easily work. I mean, he said you can easily make an oscillator like.
Is it possible that glycolysis and gluconeogenesis can happen simultaneously? Can be the correct word would be spatially and temporally related by any means?
Yes.
Like somewhere, like a seesaw, if I imagine it that way.
It has to be through an oscillator. It can happen in the same cell at the same place through an oscillator, because thermodynamically, in all calculations, you can't have it going.
Then can I correlate this idea with catabolic and anabolic processes cannot running simultaneously in a cell at a given time?
Yeah. So I personally don't like the way people define catabolic and anabolic. So for example, gluconeogenesis is an anabolic process, but it's coupled to a whole bunch of catabolic reactions. Whenever you have gluconeogenesis, it is anabolic in that you are making a new molecule, It is coupled though to precursors that are being made constantly by catabolic processes. The free amino acids are coming from polypeptide breakdown in a lot of cases, or from lipid breakdown, you're getting acetyl-CoA. Glyoxylase cycle is again an anabolic-like pathway, but it's slightly different. Same with catabolism. When you say catabolic, if you say glucose catabolism, absolutely correct. It is catabolism of glucose, but it is coupled with a whole bunch of anabolism, which is protein synthesis, ribosome biogenesis, all of that. So it is actually coupled reactions. It's the same way you think about redox. But what is important is what is the driver of the system. So in a glycolytic state, if you're calling it catabolic, you could say glucose catabolism is driving the state. The gluconeogenic state, you can say anabolic gluconeogenesis is driving this. That's how I would like to think about it.
Yeah, so I wonder, right, regarding this question of spatial separation versus temporal separation, do you have an estimate of what's the energetic cost for a cell of switching from one regime to the other? Because it seems if that's relatively high, right, then spatial separation would seem to be more optimal, maybe.
That's a really good question. We've only talked about it on the coffee table. I think though where we're going is trying to get better estimates of overall energetic cost. So having just discussed and thought about this now for a few years, I feel the way energetic costs are calculated are too crude and superficial. You have to account for a whole bunch of things over there. But the net energy gain, I think it's still that business of, how much energy is released or whatever that Jonathan had. There was the sun and there was you and something else, right? Oh yeah, so that was there, right? So we have to start thinking harder about how we calculate overall energetic costs, even for a cell. Forget the organism. I mean, the organism, there are ways to do it and people are trying to think about it. But at the level of a cell, to switch states, what is the overall energetic cost? I actually don't know. I do know though that once you switch to a glycolytic state, you're consuming way more energy per unit time. That yes, because you're anabolic in terms of building biomolecules, biomass. But the switching costs, really nice question. I think we have to start thinking about
maybe a follow-up that I also don't know is like when the cell switches, does it actually, like when enzymes are not used in this case, should we imagine that they're broken down and have to be synthesized, or they usually just deactivate?
So that's the two models, right? And that comes to time scales. If an enzyme is there and sitting there, it's not being used. There's two reasons why it's sitting there. One is it's waiting for that substrate to come in. If a bolus of the substrates come in, it's primed, ready to use all of that substrate, drive that reaction. Otherwise, it's just sitting there. But if it's just sitting there, it may be better off being broken down and used for making some other things. So now there's a lot of interest. A lot of people looking at growth and scaling and so on are looking at the costs of protein synthesis. And even more so the costs of transcripts. So any transcript is usually present at so many odd times more than the protein. In fact, there's very poor correlation for many transcripts and the total amount of protein. What is the cost of having those transcripts sitting around? Personally, I think given the collective, this is what I think, okay? So keep it as that opinion. Collective cost of making transcript is very low. Collective cost of making protein is modest, but in the scale of the whole cell, not so much. In a normal, reasonably nutrient replete environment. The only time it starts mattering is when you're in a real starvation kind of a regime where you don't have anything around. But normally for a cell, the energetic costs of making and keeping protein around or keeping transcript around is not too much, given the collective energetic costs. So approximately a yeast cell would be consuming 50,000 molecules of glucose a second. That's how much it's using. Most of it is just for ATP to do just all the cycling, making lipids, making ribosomes, all of it. Your opinion?
I was wondering too, so regarding your schematic models here, it seems that there is two scales. So there is your time scale and let's say the biochemical flux measurement scale. And in some extent to explain these equilibriums between the two populations, how can it be relevant to try maybe to scale with other level in biology? Let's say, for example, with genetics, in the extent that I am thinking when I am seeing these models with 2 cells, I'm thinking about this famous dynamic with notch delta in developmental biology. And we see that we go to the molecular component to genetics, to molecular. So there is this oscillation. And 2, also because in bioenergetics, we have few knowledge about, for example, the role that play mitochondria to regulate population, in the sense that the dynamic of fusion and fission can onset apoptosis and then regulate the number of cells in the population. So I was wondering how much it is relevant to maybe try to
I mean, I won't make too much of my model. Okay, it's a simple toy little system, but it makes us think about questions like this. I think the timescales are and how you try to break down the problem into initiating or causal bits versus things that have to follow by default. So in a way, what I'm saying here is, for example, when a when a cell has switched to the light state, it has shifted to glycolysis. Once it shifts there, all these other things will just follow. Once you shift to a glycolytic regime, everything else is so wired that you will have more ribosome biogenesis, you will make more nucleotides, you will start doing all of these. Those are all essentially consequences of the shift to this state, because you're producing more energy in that. Now, the time scales there may not be as relevant, because once you're switched, you're almost trapped in a state. Same with being stuck in gluconeogenesis, because you don't have a way to switch out of it. Once you're stuck there, you're constantly doing gluconeogenesis. So now, where is the noise in the system coming from? What are the drivers of the noise that allow you to switch states? That's a really interesting question. If you look at it this way, the only way you get noise is just small oscillations or buildups in resources. So a resource oscillator can be very powerful in driving that kind of system. These are in small to fast to medium timescales. What happens at long timescales is, if this continues, you start reinforcing gene expression programs that just get very strongly established. And if you go over very long timescales, you could have speciation type of events. Where is that? Here, where now this cell has a mutation in something. So then you now have a slightly different class of a transcription factor that's permanently on, which means your feedback, feed-forward loops, the time scales change. And the moment you have that, you have completely different cell state regimes. You are stabilizing one versus the other, or you become an obligate one type of thing. You cannot switch back to the other. That's how I would like to think about it. So a lot of these ideas on signaling gradients or resource gradients will actually scale to looking at some of these things. And of course, there is a feed forward and a feedback. Any of these will feed back and affect the dynamics of mitochondrial biogenesis, the rate of ATP synthesis, respiration. So that's just a way to start breaking down this problem. Okay.
Thanks a lot, Sunil. Thanks a lot.